Okay, welcome to the biology reporting category 1 after the bell session. One thing we're going to look at first is the assessed curriculum document. So if you go to TEA's website, or if you just Google assessed curriculum, what will come up is this page right here. And I can do that again. So I like to Google, that's easiest for me. It's usually the first one under Google. And then scroll down for biology to the assessed curriculum document and open it up. And what you have here is all the TEKs that can be assessed on the EOC outlined by reporting category. And like I said, today is reporting category 1, so we're going to look at cell structure and function along the lens of prokaryotic versus eukaryotic, mainly cellular processes, structure and function of, structures of viruses, and comparing those to cells. We'll also look at the cell cycle, and then we'll also look at cellular processes with energy conversions. So I just wanted to kind of show you this document. We're going to primarily focus on the readiness standards, and then if you do that, then a lot of the times the supporting standards are kind of bundled in there along with those. And you can see if you scroll on down, there's 5 reporting categories for biology. So the second, and I'll let you explore that on your own, but that's that document. One other thing I wanted to show you before we actually get started with looking at the stations is Lead Forward's website, where you can view the released STAR questions. So that's just Lead Forward with a 4, an actual 4.com. And if you go to STAR Resources, what's nice about this is it puts the questions in order by standard. So you don't have to go and search any longer. They're in by order by standard. So click on biology. There's lots of helpful documents on that page. This is just the one we're going to look at. So I'll scroll down. So for example, TEK B4A, this would be the question or one question that was tied to that standard, and it has the correct answer. And then it also gives you some a place where you can kind of record some data for your own class or for your own school if you'd like. And it also codes it as supporting or readiness. And then, you know, with our STAR questions or our EOC questions being dual coded, it will also identify if the question is dual coded or not. So let me show you one that's dual coded. Okay, here's one that's dual coded. It's the content standard is B.4C, and the process standard that it's dual coded with is B.3D. Are you familiar with how they're dual coded? Okay. And that's kind of all the documents I wanted to show you. I've used these 2 documents to formulate the stations that I have set up. And anything that you want as far as like the stations go, I'll email it to you. I didn't do a handout necessarily because I didn't want to make a bunch of copies when I could just email everything out so you could have it electronically. Yep. All right. Welcome to our biology after the bell session, reporting category 1. In reporting category 1, we talk about lots of different things that mainly deal with cells. So what I have here is different stations that we're going to run through today. The first station is going to cover B.4b, which is cellular processes and energy conversions, and also B.9b, which is photosynthesis and cellular respiration. Okay, first what you can have your students do is watch a short, fun, interactive song that talks about photosynthesis and cellular respiration. And as you can see here, I have a QR code. If you're not familiar with QR codes, all you have to do is download the QR reader onto a cell phone or iPod, iPhone, either way, and you read it and they can watch the video on their device, or you can play it on a smartboard or through a projector as well. Okay, next what we have the students doing is I have two different diagrams, one over photosynthesis and one over cellular respiration. What I'd like for them to do is just reference those as they are using the different equations to answer a couple of questions. And my preference would be for them to do this in groups. So maybe pairs, groups of 3 or groups of 4. But the first thing that I'd like for them to do is look at the two different equations and then answer these questions. The first, explain how energy is being conserved in each equation. So what they should be able to tell you is that they can tell that it's being conserved because you have the same amount of atoms on each side of the equation. And then also, how is energy being converted in the two equations? So what I'd like for them to do is to note that the equations are almost opposites of one another. They are cyclical. They fuel each other. So that's what they need to know for photosynthesis and cellular respiration. The last thing for this station is for the students to actually answer some questions that were on past STAR tests. And what I'd like to do is try to ask the question without giving the answer choices. So the first question is just explain the energy conversion process that occurs in the mitochondria. And I'd like for them to discuss that, explain it, maybe have them explain it on paper or to you. And then on the other side is the actual STAR question, so they could see what it may look like in a STAR situation. And then the second question for this TEK is which of the following correctly describes how a diagram of cellular respiration would differ from a diagram of photosynthesis? So notice I folded it so that the answer choices were not showing, and then as students answered that, then they could look at the potential answer choices and see which one they would choose. Okay, and in Station 2, we're going to talk about viruses versus cells and viral reproduction. This is TEK B.4c. And the first thing, again, involving our technology, have a QR code with a short video this time that describes how viruses infect our bodies and make us ill, and how they replicate inside of our bodies as well. And it's a short, I think it's about 3.5 minutes, so it's a short video that really demonstrates the process really well. The next thing I'd like for them to do is to look at the different diagrams. We have different types of viruses here, common viruses, and then we also have a prokaryotic cell and a eukaryotic cell. So using this diagram and a Venn diagram, I'd like for them to say the differences and similarities between viral structure and cellular structure. So they could fill out the Venn diagram there. And lastly, the same as before, we really want to show them what it would look like on a STAR test. And so I have a viral question here with the answer choices once again folded under. This question states, a photograph of a virus is shown below. You can see the virus here. The projections on the surface of this virus allow the virus to, and then you would want them to be able to answer that without looking at the answer choices. And if they can do that, then they really understand the concepts. And the video really discusses those viral projections and their function. Okay, in the next station, Station 3, we have the cell cycle, B.5a. And so what I'm going to have students do this time is to look at a diagram of the cell cycle with all the phases, and want them to create a mnemonic to remember the phases in order. or the stages in order. And I've also provided them an example of something they might do. This one is actually a route, but they can use the QR code once again. And this is actually student generated, so they could see what a student might produce. It's really informational. And then next, what I'd like for them to do is actually manipulate some models and the stages of mitosis. So giving them the materials here of a sheet protector with just white paper inside, an Expo marker with something to clean it with, and some pipe cleaners. What I'd like for them to do is to draw each stage of, you can do cell cycle or you can just do mitosis, it's up to you. I've provided them an example of mitosis if they kind of get stuck or need some help. And so all they're going to do is they can draw their cell on the sheet protector with the white paper, and they can show what the chromosomes would do within the cell cycle. So you can show prophase, even with the nucleus disappearing, you can show it, and then as they can erase and show metaphase as the chromosomes line up. The great thing about pipe cleaners is that they can take them apart fairly easily and show anaphase, that they're being pulled apart to the different poles. So that's a great activity for them to do. And you can have the students actually call you over after they get each phase done, and you can either mark off that they've done it correctly or have them show you the process that they underwent. Okay, the next thing on this station is to actually show them some prepared slides of what mitosis would really look like. You know, it's great to have some models of it, but sometimes it doesn't transfer to what it would look like in real life. So we've got a real life prepared slide photograph here, and what I want them to do is actually cut out the different cells and put them in order of the cell cycle. Okay. And then lastly, once again, using the released questions, and notice this has an actual photo of the cell cycle or mitosis, so it really is important that we show them what it would really look like. This one is a photomicrograph of onion root tip cell during mitosis is shown. It wants them to say which phase is occurring at the arrow. So notice I folded under the answer choices, so they should be able to tell you those without looking. And then the second question that kind of goes along with this topic is, which of these must occur during S phase of the cell cycle so that two daughter cells can be produced during M phase? Now that one is kind of, it's hard to answer without the answer choices, so I left those there. One more thing before we move on. I did want to show you more closely the cell cycle, and that you do have the G1S, G2, and then mitosis included. So it's really important that we talk about that with EOC. Okay, last but not least, we have Station 4, which is biomolecules, and this is notoriously hard for students to understand. So what I've done is I've created a graphic organizer for them, and you know, they need to know the structure and function of biomolecules. So we have the 4 different biomolecules. On top is the structure, and on bottom is the function. So all students do is flip open the graphic organizer, and they have the structure on top and the function on bottom for carbohydrates in this case, and lipids, proteins, and nucleic acids. What I want them to do with this is great information for them, but they need to be able to use it. So what I want them to do, I've cut out that graphic organizer, and I want them to play a memory game with it. So just turn them over, and they might have partners for this. They can use their graphic organizer for assistance. Turn over all the little pieces of paper like you would play memory, and then just flip over and try to match. So they're going to have to know what the structure and what the function is to be able to match the different cards together. And once they match them, of course, then they make that pair and move on. So that's for for the biomolecule structure and function. One more thing we have, an activity that we can do for this, is to actually have students generate questions. So I've left blank cards here, and I've left them the instructions of writing one question, a comparison question, between the different types of biomolecules. And for their help, I've left a few examples. Okay. Then what I'd like for them to do after they get their question made and they can use these examples, I want them to quiz each other. So almost like playing a card game where one partner chooses a card, reads the question off, and then their partner has to answer. So it's a game instead of being just, school. Okay. And then last, of course, we have our actual released questions. Both of these questions need the answer choices to be able to answer it. The first one is proteins and polysaccharides are polymers. These polymers are formed by dehydration synthesis. Which statement correctly identifies a difference in the structure of proteins and polysaccharides? So several things that they have to do in this question. One, they have to know what the structure of a protein is. They have to know what a structure of a polysaccharide is, and they have to know that a polysaccharide is actually a carbohydrate. So lots of different steps in this one question, and they have to compare the two. So that's a pretty challenging question. And then the second question for this was, which of these best represents a fatty acid molecule? So again, in multiple steps, they have to be able to identify the structure of a fatty acid, and then they have to know that a fatty acid is in fact a lipid. So the graphic organizer will really help them if they study this in knowing the difference between all the biomolecules.